Hi all. Uh, I hope the last class was understandable to you and I have not seen any doubt asked, not even from the worksheet I have given. Anyway, I have given the answers of the worksheet also. Uh, in this today, we will discuss for this week. You have a lot more things to do. That is, there are some calculation parts from genetics which is important also. They can ask plus dihybrid crossing and chromosomal theory of inheritance. So these are the main things which we are going to discuss for this week, and we have a lot of practice problems for the solutions and a few other questions also I have added. So you have to do this, and answers for this I will give you next week. Try to do it. Don't leave it that I don't know. Try 2-3 times. I know that this question is quite lengthy and it is little bit in a different manner they have given instead of giving it very directly. So try to do it. If you don't know, when I give the answer keys or the answer part next week, you can go through it, and I request is to do this. So we don't know for your examination whether they will ask these type of questions or not. Till now they have not asked, only 1 or 2 times, that only one question they have asked, not many. So they may ask. Maybe the problems from genetics are not compulsory every year, so sometimes they are asking, sometimes they are not asking, but we have to go through this. So this week we will first start with one more thing. The last page, these are some of this is the last page, that last one what I have given, that are the answers for the last week worksheet. B, A, B, like that. You have given 1, 2, 3 questions, and I have given the Venn diagram also. We will discuss. Don't confuse with this. So these are all related to this. So we will move first to the problems from blood grouping. So we know that by knowing which genes are or which alleles are responsible for the formation of blood, with that we can predict the blood group of a person. We can predict it, only prediction. So sometimes, most of the times, the prediction is right. In rare cases, prediction will get wrong, especially for this Bombay blood group, golden blood, like that. In very rare conditions it will change. Otherwise, universally this prediction is true. So we will move to the questions one by one, and these are the genotype table. You can see the genotype is given, which blood type it will form, that also given, and whether they will produce any blood proteins, that is antigen, and blood antibodies, do they produce or not, that is also given. That is, small I and I will give you type O blood group, and the blood protein, they don't produce any antigens, and they have antibodies, anti-A and anti-B. Then IA, that means homozygous, dominant homozygous, will give you type A with blood antigen A and antibody anti-B. And heterozygous, IA and small I, that is for the O, will give you A blood group with the antibody, B blood antigen A. So these table you have to keep in mind. If you don't know this table, it is very difficult to answer the questions, and these questions are mostly asked for the exams because it will come for 1 mark or 2 mark side, so they are asked in many times. So we will move to few questions. I will not explain all the questions. I have given you the answers also. These questions you can practice in different ways. It is very easy. That is why I have not given some practice problems from this, but you can go through these questions. So many of the questions are here and there asked in exams. So first question: in a patient rushed to the emergency room and has suffered severe blood loss. Type AB blood is short supply, but nurse say don't worry, his type AB positive. We can give him a kind of blood. Explain which type of AB is, why this type AB is called as universal recipient. So a person who is having, the reason is also given that the person who is having AB positive has every kind of proteins in the blood, that is antigen in the blood. There is nothing in blood type that he could be foreign into the body, so rejection is not a problem. So that is why it is considered as a universal recipient. And when you come to this third question, second question also you can read, it is related to the medics. Third question: there is a practical joker in the maternity ward who removed all the baby IDs from the bracelet. That means you know that whenever in the maternity wards in hospital, whenever a new baby was born, so at a time lot of pregnancies delivery will be taking place inside an hospital. So all these babies will be taken for washing and for cleaning purpose. So whose baby it is very difficult to identify. So whenever a female is given birth, that time itself the nurses will put a tag on their hands, and somewhere on their hands with some unique ID, name of the patient, like that. So question is related to that. There is a practical joker in the maternity ward who removed all the baby IDs bracelet. That means they have removed the tag. There are 3 babies that cannot be easily distinguished, and the parents want to be sure they get the right one, so that a doctor do a blood test. In order to find that, doctor had done a blood test. A particular mom is homozygous to type A and the dad is type O. The babies have blood type AB, A and O. Show your work below and indicate which baby must be theirs. So it was given here, the question is very clear. Parents, dad and mom. The mom is homozygous type A. Homozygous to A, not heterozygous. That means I capital A is mom, and the father is, and the parent want to be get that, and the blood test was homozygous, and dad is O, that is small I. And you see the Punnett square. They have used the technique of Punnett square. When you see here cross matching, they are getting I capital AI, that means all of them are grouping for heterozygous A. So that baby's blood group should be A, and the baby's blood group, that question was which blood type the babies will have. So it is A. So doctor can say that babies which are having the A blood type is there, they are the respective parents to that particular baby, that they can say. So these are the questions they are possibly asking. So instead of this A, here instead of this particular mom is homozygous, they can give heterozygous. Dad also, the blood group can change, but the technique to do this is you have to use this Punnett square to do this type of Punnett square. If you want to do this Punnett square, you must know O blood group genotype is what, A blood group genotype is what, B blood group genotype is what, and AB blood group genotype is what. That should be known. If it is not known, you can't find this, and this is asked for the exams. Compulsory one question they used to ask for 1 mark and 2 mark, and this can be used outside also. Many of the entrance examinations, they are asking regularly these questions, and it can be used anywhere. It is very common because it's a very direct question for me. Next one, fourth question. I will be explaining only the important ones. Other things I will not explain. You have to go through that because it is all the answers is given here. Fourth question. The police has rounded up a usual suspect in the latest rash of bookstore robbery. The thief got a nasty paper cut at the scene of the crime. The suspect are of blood O, A, B, AB. The blood at the crime scene contained one I allele, which suspect therefore cannot be involved. Explain. That is a question. Is it clear? That means the blood at the crime scene contain I allele. So which suspect therefore cannot be that means the blood at the crime scene contained an I allele. Full stop. Which suspect therefore cannot be involved. So which is the suspect that cannot be involved in it? So that means suspect number 4 should be removed because they have completely dominated IA, the heterozygous, co-dominant in nature. Balance all are having I inside that, so they can be taken. So these type of questions they will ask. So please go through this, and if there is any doubt, you can ask me. And in the 9th question, you see, the father of 2 children is type O positive and the mother is type A positive. The children are O negative and A positive. Given this information, what can you say that the genotypes of father and mother? That positive and negative is the Rh blood group, that is rhesus factor, which you have studied in +1 in circulatory system. You have studied about it, and some diseases also you have conditions also you have studied there. So that is a question, and the question they have not said whether homozygous, heterozygous, nothing they have said. They are directly asked what should be the genotype of father and the mother, and you see the answer. If the father is O positive, his ABO genotype is II, that is common. He may be either Rh positive or heterozygous, that means homozygous to positive or heterozygous to the Rh. If the mother is A positive and her blood group genotype should be homozygous A or heterozygous A, and her Rh should be either ++ or +-. However, either of the children is Rh negative, because here either of the children is Rh negative. Both the parents must be a heterozygous. In order to get a negative child, each parent should have. I think you have understood from that. If you want to get a negative child, so both the parents should be negative. If one of the child of the O, then the mother must be IAI, which otherwise the child only will be type A. That is clear from that. So the father ABO blood group is O. Rh type should be positive or negative, and the mother ABO blood is IAI, and Rh should be positive or negative. So it can be mixed, so that this Rh gene can be expressed. This 9th question is not asked very much, but keep in mind this can also be asked. Clear? Then 10th question, that is, how many types of gametes would be formed by selfing capital A, small a, capital B, capital B, capital C, capital C, capital D, capital D. So the direct thing what you can do is, if you can count how many heterozygous are there, you can get how many different types of gametes can be formed. That's a direct question. Other direct answer you can see. So here there is only one heterozygous. So the formula is 2 to the power n, that is 2 raised to n. 2 stands for the two gamete, that means the two basic gametes from which they are forming, and n is the total number of heterozygous. So here n is 1 because we have only one heterozygous. So the gametes possible to form is only of 2 to the power 1, that is only 2 gametes can be formed. So what are these gametes? So here that picture of the gametes are not coming properly, so I have given this gamete is here. How we are getting the 2 gametes? Excuse me. Yeah, here one. So just to see that it was capital A, small a, and all other were capital letters, capital B, capital C. That means A can mix with B and B, C and C, B can mix like that. So we will be getting ABCD, 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 ABCD. That means all will be ABCD, capital A, capital B, capital C. So that will be one gamete from that. And here small a can mix with B, capital B, capital B, capital C, like the capital D. So it can give small a, B, C, D, like the 2 gametes can be formed. So this question is asked. So this question can be asked in many forms. I have given some problems in this question paper, so you can do according to this. Is it clear? Or any doubt, you can put a message wherever I am posting this video. No issue for that. I will revert back. Is it clear? And next comes, that is coming is the probabilities in genetics. So with the probabilities, there can be 1 or 2 questions can be asked. That means whenever we are doing this Punnett square, this Punnett square can be done only in a condition where there is only a small data are available. That means 1 or 2 crossing, not big crossings. Punnett square cannot be used. So in order to do that, we will be using the basic rules in probability. One is the product rule and another one is the sum rule. That means multiplication and addition rule. So you can read the data, but here I have given one example. Suppose a mother and father is both heterozygous for a characteristics of brown eye and a brown hair. That is, they have brown eye and brown hair, but carry the recessive gene for a blonde hair and a blue eye. Calculate the probability that they would produce a blue-eyed, blonde-haired boy as a child. This is the question. So the answer you can see, we have something to do, then you will understand more. Since one gene from each parent is given for the character trait, together with the sex determination can be carried by the 23rd. So these are data. Okay, we will move to this. This you may not understand very clearly. So we will move to this question. That is question number 7. Question number 7. You see there, let us cross capital A, small a, capital B, capital C, small c, capital D, small e. So this genotype is given, this parental genotype is given, and they have asked a question: what is the chance that a particular offspring will be having this particular genotype, that is capital A, small a, capital B, small b, capital C, small c, small capital D, capital E, capital D, capital E, small e, like that. how this offspring will be having this particular type of genotype. This is what we have to find. Parental genotype is given. From the parental genotype we have to know whether any of the offsprings will be having this type of genotype. So here if you see, one thing you can understand, two different characters from the parents, they are mutually understanding and coming together. So whenever these type are coming, we will be doing multiplication. And if a question is coming, that is from this anyone, that means from a family, how many will be a boy, how many will be a girl, like that we will be doing the addition. This is the basic rule. So you can understand that two different characters, that means two different traits mixing together, and they are giving a one genotype. We are going to find out whether that parental genotype, when they are mixing, we will get this particular offspring in this genotype. So in this condition we will be using the multiplication rule. So how we can do this multiplication rule? With the help of multiplication rule, how we can do this? So how we can do this, I have written here with pen, because when we are doing the Punnett square it will be very difficult. So the cross is given AABB, and if you see here, you can see from the two parental sides, one thing is very clear. That means they are almost carrying the same genotype, whether some are homozygous, some are heterozygous. That means same alleles for one character they are carrying. It is in some are in heterozygous, some are in homozygous in nature. So we can divide it into a monohybrid crossing, and from each monohybrid crossing we will see whether that particular genotype is forming. If that genotype is forming, finally we will get out of this much offspring is possible to get this type of genotype. That is the basic thing. And if you see here, I have divided from the parental side. First we have heterozygous A, capital A and small a, and from the consider the first one as male and second one as female. So A and H is there, and I have divided it into a monohybrid crossing by using the small Punnett square. And here and check with the genotype, offspring genotype. Here they are asked whether the offspring will give or get a heterozygous cap A. Yes, chance is 1. Sorry, chance is 2. Two chances they can get, that is heterozygous. And next one is B. So here also the chance is 2. And the next comes is C, and here they are asking small c. Small c is also possible 2 times. And next they are asked this capital D is possible, that is for 1 time. And the next they are asked is heterozygous E, that is capital E small e. Heterozygous E is not possible because from the parental side both are homozygous E, capital E small e. So when we multiply, there is no. So since we are doing multiplication, we will get 0 there. So any number multiplied with 0 is 0. Because of this reason, there is not possible to get any offspring with capital E. So this genotype is not possible. Am I clear? And here when I asked it should be it is not heterozygous E, it should be capital E, capital F and capital F. So please clear that. Is it clear? Next question that is coming is, that is the sum rule. So where this sum rule is applicable, so we will see one example from that also, and we will move forward. Here is it. 8th question. What is the probability that all 3 children in a family will be the same gender? That's a question. This is also a genetic question. This can be also asked for the exam, and the 9th and 10th you have to solve by yourself by understanding this. So read the question carefully. What is the probability that all 3 children in a family will be the same gender? Clear? So this here they have gone with the set rule. Both are right. Whether you are done in another way or set rule, both are right. I feel the set rule is easy. That means, what is the chance to get all females child? You consider in a human way. So in one fertilization there is a chance of only one sex, either male or female. Clear? That means 1/2 from the 2 sex, from the male or female, any one sex is possible. So like that, 1/2 total 1/8. That means in 8 child, 1 child is probably a female. Same way for male also, same thing. From the 8 child, 1 child will be of male. Next one, see here, all one gender. So when they are coming, when this question is coming like that, we have to add both male and female together. That means out of 4, there's a chance of getting the same gender. Out of 4 there's a chance of getting the same gender. That is why they have done addition here. Is it clear? Another question is also, 9th question is also the question number mistake. The 9th question is also the same question with another one. What is the probability that the 3 child is 2 girl and 1 boy? So that also you can go through that, how it is. That means from a female, you understand this thing very carefully. That means chances of getting female in a 3 children, it is 1/8 chance. All the 3 children in a family will be the same gender. That is female, it is 1/8 chance. For male also it is a 1/8 chance. All the 3 children in the family will be same gender. That means if you are taking first child as male, second child as male, third child as male, how many times, how is it possible to get? Next, female, like that, is it possible to get? That means they are asking any one of this, or they are not asking and. So there we are asking 1/8 added. So wherever this or is coming, we will use addition rule. Wherever and is coming, both together, capital A. If you see the top question, you can understand that 2 different traits together they are coming. There we will be using the multiplication rule. So you have to do this question with understanding, because I don't have that facility to teach on the board. I have tried, but the clarity when we are taking this is not coming properly. That made me to explain like this. So any doubt, please contact me. I will explain you. I will find out a way to explain. Is it clear? Next we will move to the, all this you have to do. It is a sentence type question. Next week I will give you the answer for this. I have the answers. I want you people to try, all so that the concept will be clear for the exams. And this thing also, please don't forget, so you can see on this. So next one, we will move to dihybrid crossing and the chromosomal theory of inheritance. So here in dihybrid crossing, last week we had discussed about the monohybrid crossing, that means single character. So here the Mendel has taken two characters at a time. That means if we are mixing, if we want two different characters, if we have taken, what will be the result? Is it the result according to the monohybrid crossing, or it is some other thing? That is what the Mendel was trying to see. So when the Mendel did this, you are already little bit you have studied this in the 10th standard. The rules are same, there is no change. That means whenever the two characters are taken as male and female, the characters have the capacity, that means genes will independently assort. Assort means they will mix independently. They will not see whether it is mixing with the green is good or with the yellow is good. They will mix independently. So this is what the Mendel was proved from the second law of inheritance. That means a dihybrid crossing. So this type of mixing of genes are also taking place in us, because whenever the meiosis are forming in the gametes, especially in the gametes, through meiosis, when the gametes are formed, so the genetic material, we have different characters in us. Some are dominant, some are recessive for this, that, etc. It is so in our gametes also, formation of gametes also, law of independent assortment will be formed. Law of dominance and law of segregation will be less, but maximum it is a law of independent assortment. This was the Mendel proved from the dihybrid crossing. And in F2 generation, he got a sequence as 9:3:3:1. That was a phenotypic ratio he got. So that's all from this Mendelian dihybrid crossing. So in your textbook they have given it that crossing. You can use same thing, or you can use any of the Mendelian traits. That means Mendel has used 7 different parts as an experimental material from the pea plant. So any of these 7 parts you can mix and take. So in the textbook they have given as a seed green, yellow, like that, round and wrinkled. But if you see here they have taken the seed pod. So whatever you take, the answer will be the same. Answer will be the same, 9:3:3:1. Is it clear? And all this thing, whatever I have said, that also how the gametes are independently formed, the rule of Mendelian inheritance, independent assortment is forming. And some questions I have given here, please go through that. Though they all are easy questions, they are not difficult questions. And the last part for this week that we have, that is the chromosomal theory of inheritance, because the chromosomal theory of inheritance is what made the scientist to found out what Gregor Mendel did. This was the beginning. So since Mendel did that experiment first, that was put first, and the chromosomal theory of inheritance was put second. But in 1900s, when the people started to study, they started to study the chromosomes. Through that they found out Mendel. So here in chromosomal theory of inheritance, 3 scientists, that is Walter Sutton, Theodore Boveri, and Thomas Hunt Morgan. Why Thomas Hunt Morgan is given here? Because whatever the chromosomal theory of inheritance, what Theodore Sutton and Boveri has said, Thomas Hunt Morgan is confirmed it through his experiment, through the Drosophila, which comes in linkage and recombination, that we will study next week. So that is also a big topic to explain. So they have found out whatever the Walter and Theodore Boveri and Sutton said, Thomas Hunt Morgan is confirmed it. That's all. And he found out a new thing that is known as linkage and recombination. So he is the first person to say that all the genes are not independently located. That means independently on a locus. Some of the genes are linked and some of the genes are non-linked. So when they are linked, they will express like this. When they are non-linked, they will express, and they talk about the recombination. That means the mixing of crossing over, meiosis crossing over. That is mixing of the genes. So that is why Thomas Hunt Morgan was given here. Walter and Sutton and Theodore Boveri, they are the two main persons who said about the chromosomal theory of inheritance. So in chromosomal theory of inheritance, the concept is this. That means whenever the gametes are forming through meiosis, either the same chromosomes will form as gametes. That means the chromosomes will be divided, two will be divided into 1 and 1. Each have the capacity for the mixing. They will independently done after the meiosis, or otherwise in meiosis 2 they will cross over each other, and it may give a different rate. That's all the chromosomal theory of inheritance is. Am I clear? That means the chromosomes are responsible for these variations, and inside the chromosomes, what are there? Genes. And genes are what? They are the DNA. They are the DNA. That means the chemical unit that is responsible for this change, that is DNA. And these chemical units are present in the form of genes, the unit of inheritance, and it is all many genes together makes one chromosome. So if that one single stranded chromosome is coming from the parent and forming into meiosis, through meiosis, and the same chromosomes are there, so the almost the parental character will be same parental character we can see in the offspring. Sometimes during the time of meiosis, these chromosomes may cross over. When the crossing over of the chromosomes takes place, there can be a chance of variation that causes the variation. That is what these chromosomal theory of inheritance means, and this was confirmed by Thomas Hunt Morgan. We will discuss this in next class. So forgive me for putting this like this, because I tried with the blackboard, because the blackboard, when I was taking the video, it was not coming in a good quality. That made me to explain mathematical part like this. So please forgive me. If there is any doubt, kindly message to me. You can take a number from the father, or if you need my number, just put a message. I will give my number. You can contact me through WhatsApp or you can call me. No issue. Am I clear? Thank you.